16. Get this, oops, sorry about that. Get this rolling. So chapter 16 is called The Molecular Basis of Inheritance, part 1. So in this chapter, we're going to focus in on what is the structure of DNA and how is it replicated, and then a few other small topics. By the way, these guys right here are Watson and Crick. Watson and Crick won the Nobel Prize for determining the three-dimensional structure of DNA. They, of course, had help from lots of other people, including probably someone you know or have heard of, Rosalind Franklin. Anyway, let's move on. So there's a big section of history in this chapter at the beginning of the textbook. I just want you to think about, I just want you to know a couple of names. This is one name that I think you want to know. Now let's go back to the early to mid 1900s, and it really took this long. In the early 1900s, people started realizing that there's this genetic something is controlling the behavior of our cells, and what kind of molecule is the heritable bit of information that we pass along. And researchers thought it was proteins, because proteins are really complicated. All proteins have unique three-dimensional shapes. They can be really complex, and it seems like genetic information is pretty complex. Maybe it's proteins that are the heritable structure, or the heritable molecule. And it turns out, of course, it wasn't, but there were a number of research experiments to kind of identify that it's DNA that's the genetic material, not proteins or something else. And so Chargaff in the 1940s was one of the people who was in the thick of things. And Chargaff noticed a couple of things that are worth pointing out, and this is something you surely know already. But here we go. Chargaff was investigating DNA composition in many different species. And so he started looking at, the percent of each base, each nucleotide, if you will, in each species. And he realized that they vary across different species. So that's number 2. The proportion of total DNA made up by each base varies from species to species. I mean, if you look at humans versus oxes or salmon or sea urchins, the percent of adenine varies. But he noticed, and this is number 1 here, something really interesting, that the percent of A, or adenine, always is very close to the percent of thymine in every species he looked at. And the percent of guanine is very close to the percent of cytosine in every species that he looked at. So he made what we call now the Chargaff's rule, that the percent of A equals T and the percent of C equals G in all species. So that's something that they were finding out when there was a big race to try to figure out the structure and the replication of DNA. Now in the 1940s, researchers had already understood at least some things about the structure. They knew, for example, that DNA is a polymer, and it's made up of nucleotides. So we'll want to fill in that blank right there with the word nucleotides. Oh, my phone's ringing. I'm sure it's my mom. She's the only one who calls me on my landline, but I'll have someone else pick it up. And then we might ask, what are the 3 components of a nucleotide? Because each nucleotide has 3 things in it. The first thing that it has, well, they're all together, is phosphate group. I should have just written this one as a P with a circle around it, right? A phosphate group. Every single nucleotide has that. Every single nucleotide has attached to that phosphate group a 5-carbon sugar. So think of like deoxyribose or ribose. Those are 5-carbon sugars. They form a ring. So we have a phosphate attached to the sugar, and that's attached to a nitrogenous base. You're going to have to spell this one on your own. Nitrogenous. Let's add the O-U-S there. Base. Okay? A nitrogenous base. So let's take a look at a picture of a nucleotide and several nucleotides put together on the next slide. Okay, so this is a picture of a short nucleic acid, because that's what DNA is, a nucleic acid made up of, this one happens to have 4 nucleotides, all attached to each other. Each nucleotide, you can see here, one of them is highlighted in this little box. Each nucleotide has a sugar. DNA has deoxyribose. Attached to the 5th carbon, we call it the 5' carbon of the sugar, is the phosphate group. Attached to the 1st carbon of the sugar is a base, a nitrogenous base. There are different bases. Okay, 2 of the bases have a double ring structure, that's guanine and adenine, and 2 of the bases are smaller, thymine and cytosine. And this is a picture of 4 nucleotides put together, and you can see then that the nucleotides are held together here, connected with covalent bonds at the 3' carbon of the sugar. So we can see that every nucleotide becomes attached at that same location. And when we look at the whole nucleotide chain, or the polymer, if you will, it has sidedness to it. So up here, we call this the 5' end because the phosphate from the 5th carbon on the sugar is sticking out. Down here, we call this the 3' end of the molecule because the hydroxyl group on the 3rd carbon of the sugar is sticking out. That's kind of the end of the molecule. So we would say that this molecule has polarity. It runs from 5' to 3' is often the language we use. Okay, this doesn't look exactly like DNA, though, does it? It's at this point in history, people knew that DNA was made up of nucleotides strung together somehow, but they didn't quite understand how the 3-dimensional shape of DNA. So they had to figure it out, and there was this massive race going on in the 40s and early 50s to sort out the 3-dimensional shape of DNA, or the structure of DNA. So here we're thinking about, what does it look like? What is its 3-dimensional shape? Well, obviously we know it's a double helix, right, made up of 2 strands that are interwoven, but they didn't know that. Some researchers thought it was made up of 3 strands, somehow worked together. Other researchers couldn't figure out how the double helix worked, or how the 2 strands maybe fit together. And finally, in the 1950s, Watson and Crick, who were working in England, Watson was an American researcher, they published a paper in Nature that definitively said DNA, that basically stated a number of different pieces of the puzzle, and basically they elucidated, or they figured out the structure of DNA. And they received the Nobel Prize for doing so. But I do want to say that there were other people involved in this, and one person who is now getting credit for her work but didn't then was Rosalind Franklin. Rosalind Franklin was a woman scientist in the 1940s. This is a tough place to be, and she worked in England, and she worked with a researcher named worked with another student named Morris Wilkins, and she was basically a really good chemist. And she worked in a lab in which she was studying DNA by making DNA into crystals. This just eludes me. I'm not exactly sure what she was doing, but she was doing and then she was performing what's called X-ray crystallography on the DNA itself. She was taking these X-rays of this DNA, and by looking at the pictures, or the X-ray crystallographies of the DNA, she could sort out all sorts of things about the structure of DNA. She had figured out it was double-stranded. She had figured out the the phosphate and the sugars were on the outside of the molecule. She hadn't quite sorted out how the bases paired together yet, but she had figured out the diameter of the molecule, and she was essentially pushed back and told, she was a woman, she couldn't go into the places she couldn't go to like the lunch place with the men because she was a woman. She was kind of poo-pooed by Watson and Crick, who worked in a different university, although they at one point sort of received from one of Franklin's colleagues who worked with her and didn't really like her. His name was Maurice Wilkins. He gave Watson and Crick a picture, DNA of Franklin's X-ray crystallography of DNA, and she didn't know it. Watson and Crick used that picture to figure out how DNA is organized. It's three-dimensional structure, and then they didn't give her full credit. So it's an interesting story if you've ever want to read more. This is the most recent book on Rosalind Franklin, or one of the most recent books on Rosalind Franklin, and it's very good. The Dark Lady of DNA. By the way, she died before she could be awarded the Nobel Prize. They don't award Nobel Prizes after someone dies, and she died of cancer. And I guess that's not a surprise because she worked with X-rays all the time back in the 40s. You didn't really take protection. But either way, she died of cancer at a very, very early age. She was phenomenal at what she did. Okay, so let's look at the three-dimensional shape of DNA as Watson and Crick outlined it way back in the 1950s. So they said, all right, first thing we want to tell you is DNA is double-stranded. So let's put double-stranded in here. I might not write everything down because this takes like a lot of effort. Okay, DNA is double-stranded. The two strands run in opposite directions. They run anti-parallel to each other, okay? Anti-parallel to each other. So if we look at, I'll do a drawing here, but yeah, let's do a drawing. So if we're looking at two strands of DNA, one strand is running 5' to 3' with that phosphate group sticking out over here, right? The other strand is running 3' to 5'. They run anti-parallel to each other, opposite directions, okay? And then the next thing to note are the next 2 points on that last slide, okay, is that the outer, this sort of looks like a ladder, right? The outer edges of the ladder are made up of phosphate and sugars. So let's go back up. If you look at this picture in the middle here, the phosphate, sugar, those form the outer edges of the helix, if you will. So you can fit that in here. And inside the double helix then, and inside the double helix is the bases, the nitrogenous bases. They form the rungs of the ladder, the inside of the helix, and in fact, they pair together in a particular way, all right? They pair together, and this is going back to Chargaff's rule. It now makes sense. So that adenine and thymine always meet up, and hydrogen bonds form between them. So adenine is hydrogen bonded to the thymine of 2 adjacent nucleotides. And cytosine and guanine always pair up. This time they form 3 hydrogen bonds between them, if you can see that. But C and G always pair together. So nitrogenous bases pair together, and we call this complementation or complementarity. What did I use in my complementarity? Not complete. Complementarity. Ooh, that's barely going to fit in there. Complementarity. Okay. Let's draw that in. So if we have an A here, well, maybe I should change the color of my pen. I don't think I can. I'm stuck with this color. Oh, no. I just want to have a different color so you can see it. If we put a G here, there's going to be an A here. Or if I have a T, and another T, and a C, and an A, and a C, and a T. You can figure out the base pair sequence of the alternate strand, if you will. So if there's a T, why did I put it? Oh, wow, did I make a mistake? This should be a C, right? G pairs with C, T pairs with A, C pairs with G, A pairs with T, C pairs with G, T pairs with A, like that. Yeah, you can figure it out. And Watson and Crick actually sorted out that the A and the T always pair together and the C and the G always pair together. They actually used some of Rosalind Franklin's work. Rosalind Franklin said, hey, here's the diameter of a molecule of DNA. She knew the diameter. They used that diameter to sort of Goldilocks their way through the three-dimensional structure of DNA. By Goldilocks their way, I mean they thought, if there are two strands running side by side, and the strands are only this wide. So if we put two purines, that's the A and the G, together, then they'd be too big. The strand would be too wide. That'd be too big. If we put two pyrimidines together, that's the T and the C, then the strand would be too narrow. But aha, we've found it just right. We have one purine and one pyrimidine. And now we've got the perfect width for this molecule of DNA. So they discovered the three-dimensional shape of DNA, along with getting help from a lot of other people along the way. But DNA is made up of a double helix, right? The two strands run in opposite directions. We can see here that this strand has the 5' end up here. Its partner has the 3' end. The sugar and phosphates form the outside. The nitrogenous bases form the inside. Okay? And they pair together in a relationship called complementarity. If you really want to put that in your notes, A equals T, C equals G. They pair together, all the time. Okay. In 1953, Watson and Crick published that article. It was a one-page article in a publication called Nature. And Nature is a very impressive journal for science. But they wanted to make it clear that they not only did they understand the three-dimensional shape of DNA, they also probably could guess how DNA replicated itself. And so at the very end of their paper, they wrote one sentence. This is the very last sentence of their paper, and I just think it's worth thinking about. It has not escaped our notice that the specific pairing we have postulated, and by that A pairs with T, C pairs with G, immediately suggests a possible copying mechanism for the genetic material. If we know the nucleotide sequence of one of the two strands, we can determine the nucleotide sequence of the other strand. We just did that on the last slide. Watson and Crick figured that's how DNA replicates. It's called semi-conservative replication, and they were right. Finally, in 1958, Michelson installed it, kind of a definitive experiment to demonstrate that DNA replicates in a semi-conservative model or mechanism. Let's show you what I mean by that. So if you've got a molecule of DNA made up of two strands that are wound around each other, right? This is the old DNA. What happens in semi-conservative replication is that the two strands separate from each other. Okay? And then each strand becomes the template for the building of a new strand. And this is when I have to use my different colors here. Okay? So that this old strand here is used as a template for the building of the new strand of DNA, because if we know what the old strand's nucleotide sequence is, we can build a new strand of DNA. The second old strand becomes the template for the building of another strand of DNA. And so what we're left with when we replicate our first strand of DNA is 2 copies of DNA that are identical to each other. They have the same nucleotide sequence, but they each are composed of 1 old strand of DNA and 1 new strand. And so we say, aha, DNA replication is semi-conserved or semi-conservative, because semi meaning each new molecule of DNA has 1 old strand and 1 new strand. It's semi-conserved. Okay. Here's another picture of that, just so we know what we're talking about. If this is our parent DNA molecule, or a tiny little piece of it, the 2 strands are going to separate from each other, and then each strand is going to be used to build a new complementary strand. So when we see an A, hey, put in a T. When we see a C, put in a G. Put in an A, put in a T, put in a C. And this strand will then put in an A, put in a C. Oops, that was a mistake. Put in a T, put in an A, put in a G, right? I did not need that. And we end up with 2 molecules that are identical to each other. Yes. So how is replication achieved? This is part 2. So this is going to be kind of the bulk of how DNA is replicated. That's our next video. I'm going to stop right here, and we will move on. Okay.